So the thing we introduced in this particular, or we were talking about at the end of the last teaching portion, is this concept of this term exergonic and endergonic. They are very many times coupled, and it is what happens when we make ATP. What you have on the left with your exergonic is you have reactants. Now the column is large not because it's reactants. The column is large because it represents a huge potential energy. Those reactants have a lot of energy built into them, and they are going to go to a lesser form of energy, so they're going to go downhill. That then is going to release energy. That's this concept of an exergonic reaction that releases energy. Now that energy will then be able to be used to actually run an endergonic reaction, where you have reactants. Those reactants don't have to be the same as the products from the exergonic, but could be very similar to products that are certainly the energy level, the potential energy level of the reactants in an endergonic reaction start out very low comparatively. And then energy is put into those reactants to create products. What we're talking about here is exergonic, otherwise known as catabolic, breaking down, and endergonic, otherwise known as anabolic pathways, building up. Those here you see the terms. Exergonic reaction in boldface proceeds with a net release of free energy, that delta G, and exergonic absorbs free energy from its surroundings, and again tend to work in a coupled way in terms of building up, and in terms of breaking down food, then building up more of yourself. With regard to a closed system, we said this before, eventually equilibrium is going to be reached and no more work will be done. Open systems, constant flow of materials. There's constant flow of energy in an open system, and biology is an open system. The reactions, here you have an image of the reactions in a closed system. What we're using is a hydroelectric system as our example. What's going to happen is the water is going to turn the wheel of a hydroelectric system only until the water reaches equilibrium. Then the wheel will no longer turn. Something has to come and actually pump the water back up again so that it can turn the wheel. But in an open hydroelectric system, the water is going to constantly go down and then be brought back up so that it goes into the first tank. You can see that the first tank, the spigot's on, the water's going in, and then the water is going to turn each of those turbines and create energy. This particular slide repeats what we've kind of been saying, but it's always nice to repeat to get things in our head. Cells are not in equilibrium. They are striving for equilibrium. They're an open system. Flow of materials in and out of the cell prevents it from ever reaching equilibrium. And understand the metabolism is more like a multi-step open system. So metabolism is when we talk about it in detail. And remember, this particular is Intro to Metabolism, where we're learning the concepts of metabolism, but certainly the electron transport chain is going to be a multi-step system. We talk about the electron transport chain next chapter. ATP is our big molecule, and I think I've probably started out the semester talking about ATP. So we've talked about ATP the whole semester so far. Works by coupling these two types of reactions, exergonic and endergonic. And ATP does three main kinds of work. It does chemical work, So the pushing of endergonic reactions, which would not occur spontaneously. So ATP creates the activation energy needed for these endergonic reactions. Transport work by pumping substances across membranes. So the transport kind of causing that turbine to turn so that something can move from point A to point B. And then mechanical work such as contracting of muscles, beating of cilia, beating of flagella types of work. And you have the three images here. Mechanical work, what you're looking at is your motor proteins. With transport work, you have these proteins that are embedded in the biphospholipid plasma membrane, and then the building up, chemical work, the building up of molecules from substrates. There's the term ATP, adenosine triphosphate. It's what's known as the body's energy shuttle. It's composed of a ribose, that's a sugar, an adenine, which is a nitrogenous base. Those are the same types of bases that make up your DNA, and 3 phosphate groups. The bonds between the phosphate groups on the tail are what's broken down in hydrolysis, broken down, given off energy, and the energy comes from the chemical change to a state of lower free energy, not from the bonds themselves, but the 3 phosphates on the ribose and nitrogenous base is a very unstable molecule. It's high in potential energy because of its instability. It wants to go to a lower state of energy, more randomness of energy, and so that phosphate gets released. And by the way, there isn't an animation. Sorry about that. The chemical how ATP performs work, in the cell, in the energy from the exergonic reaction of ATP hydrolysis, so breaking off that 3rd phosphate, can drive endergonic reactions by using this concept of coupling. Coupling reactions, an exergonic reaction releases energy. That energy then is put into a reaction that is non-spontaneous so that it can proceed forward. We consider that an endergonic reaction. There is a new term that's being introduced on this slide. So the rest of this is just review of what we've been saying, is this phosphorylation. This concept of phosphorylation, which we'll use this word quite a bit in chapter 9, is this idea of transferring a phosphate group to some other molecule. So putting a phosphate group on ADP, ADP, so diphosphate, becomes phosphorylized into ATP. The recipient molecule, which would be ATP, is phosphorylated. And again, what I've just said, catabolic pathways drive the regeneration of ATP from ADP and phosphate. So catabolic is breaking down. We also anabolically build up. The energy to phosphorylate ADP comes from somewhere else. So comes from a catabolic reaction somewhere else to cause that phosphate to be put onto ADP. So the putting of the phosphate on ADP to make ATP, now please understand that that's an anabolic reaction, but the energy to do that comes from somewhere else, comes from a catabolic pathway. ATP is our renewable source of energy because of that instability, that phosphate group. And even the act of making ATP, or energy currency, couples the exergonic and endogonic reactions. New concept for this chapter anyway, but not a new concept to any of you, is this idea of an enzyme. It's going to, we just talked about the amount of energy needed to run a reaction. Now we're going to help that out. We're helping that out with enzymes. What enzymes do is speed up the reaction by lowering the amount of energy for that's needed for that reaction to go forward. And enzymes are catalysts. They are an agent that speeds up the reaction without being consumed in the reaction themselves, so they live to perform another reaction. Enzymes, a protein, and here you have an example of an enzyme helping to make sucrase. So you have sucrose, C12H22O11, and what we're going to do is break the bond there, that oxygen bond. We're going to hydrolyze, we're going to put water in and form glucose and fructose. So we are actually causing a catabolic behavior, a catabolic reaction. That catabolic reaction then is going to release energy that then can be used elsewhere. And there you have, and note this, the substrate is sucrose. An enzyme is sucrase. So what you have is ASE holds to our idea that enzymes end in ASE. And the enzymes, what they do is they reduce or lower the activation needed and activation energy needed. And so every chemical reaction it involves bonding and breaking the bonds. The initial energy needed to start a chemical reaction is called the activation energy, and it's written large capital E small a. That's that whenever we see that capital E small a, we think of reaction activation energy. And I'm sorry if I said that incorrect. So free energy of activation, or activation energy, is abbreviated as capital E small a. And every reaction that happens in the body has an activation energy. The catalysts lower that, the enzymes or catalysts lower that activation energy. They don't affect the change in free energy. Instead they will hurry up reactions or they will hurry up, or they facilitate reactions that would occur eventually. So please understand that the thing that's being said here is enzymes don't make a reaction take place. That reaction would happen anyway. So my car is going to rust, but if I don't keep it clean, if I keep all that salt on it, if I keep all the grime on it, that rust is going to happen faster. And in the body, this happens. This is true also. You are going to make ATP. You're just not going to make very much without the help of these enzymes. So enzymes are constantly doing this. They are speeding up the reaction, and they're speeding it up by lowering the activation energy, and but they aren't creating any reactions that wouldn't already take place. They are specific, and they are substrate specific. So enzymes, one enzyme will work in a given reaction, but that may be the only reaction they work on. So they are specific to one reaction, and one of the reasons why they are is they have an activation site. On this diagram, you have a kind of a pocket or groove in the enzyme. We're looking at the blue, and I need to move on to the next teaching time.